In this video, we will discuss about the functioning of the hydrogel, whose different characteristics we have discussed in past few videos. So once they have defined their mechanical characteristics, and now they wanted to check whether their hydrogels can activate the T cells and the signaling and their corresponding signaling pathways or not. So they microprint the hydrogels with different size of the anti-CD8 plus T cells, and what they have find out is that with 8 micrometer diameter T cells they are able to see a stable T cells attachment, and at the same time what they find out is that the concentration of the T cells that can give a proper attachment is 100 microgram per ml. So these are two conditions that they have optimized. That is, the microprint size of the anti-CD3 plus is 8 micrometer in diameter, and the concentration of T cells for the binding is 100 microgram per ml. So they bind this Jurkat T cells for 15 minutes, and after that they wash the cells and check the cells in the microscope, and they are able to see that at different stiffness they are able to see the binding of these T cells to the Jurkat, binding of the T cells at the anti-CD3 microprinted spot on the hydrogels. And in order to check whether this T cells activation works or not, they check the Ca2 plus measurements, which will get initiated when the T cell receptors interact with the anti-CD3. So what they have find out is at different stiffness they see the Ca2 plus levels. For the 2 kilopascals they do not see an increase in the T cells Ca2 plus signals, but for this 12 kilopascals and 50 kilopascals they can see the continuous increase in the Ca2 plus signals, and this indicates that the T cells is bind with the anti-CD3 receptors that are microprinted on the hydrogels that will cause the T cells activations. Once they have validated the T cells activations through Ca2 plus levels, they also need to check whether it will trigger the early T cells activation signaling pathways or not. So they check the two of them common phenomena that observed in the early T cells activation, that is F-Actin movement to the plasma membrane, at the same time ZAP70 also movement to the plasma membrane. And and in this case what they did is they stain this F-Actin using this green fluorescence and ZAP70 phosphorylations using the red fluorescence. ZAP70 with the green fluorescence and F-Actin phosphorylations with the red fluorescence. So what happens is that when the T cells get activated, these F-Actins will starts to move in the periphery of the cells or near the plasma membrane, and this is what they have observed at different stiffness of the hydrogels, that the F-Actins starts to move to the periphery of the cells. At the same time, they also see the ZAP70s in the resting conditions. ZAP70s are present in overall cytoplasm, but once they get activated, they move close to the periphery of the cells. So this is the ZAP70 signals on the panel A, on the left hand side of the panel A, and the right hand side of the panel A, which it shows a quantitative analysis of the presence of F-Actin and ZAP70 at different at relative distance from the within the cells. And what they find out is that on the periphery the amount of F-Actins is higher, and whereas the ZAP70s are commonly uniformly expressed and different stiffness, and there is not much difference that has been observed at different stiffness of the hydrogels about the ZAP70 phosphorylations, but they have seen a clear difference of this F-Actins movement in all the three cases. That is, they are moved towards the periphery of the cells or in the near the plasma membrane. and these things are further quantified in the panel D, where we can see that the spreading area of these ZAP70s are uniform. They do not get distributed. But for the F-Actins, we can see slight changes, but the spreadness is slightly changed, but this change is not statistically significant. So we can say that for both the F-Actins and ZAP70s, they have observed the changes, but the changes are not statistically significant at different stiffness of the hydrogels. So from these two experiments, they able to show that the T cells activations occurs on the microprinted anti-CD3 proteins on their modified or on their hydrogels, on their newly developed hydrogels at different stiffness. So I hope you understood their experiments that shows that their hydrogels is their hydrogels developed mechanism is able to activate T cells with the anti-CD3. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.